Welcome back to the second video of the cell division series. This is Bio Tan, the host of this series. In our previous video, we learned several things. We learned number one about the genetic code, which is like a recipe book that is read in triplets of bases, known as codons, and a series of these codons translates into a series of amino acids, which is essentially the primary structure of a protein. So a sequence of DNA that codes for protein, we call it a gene. In today's video, we'll be expanding on the concept of genes and how this relates to homologous chromosomes. But before that, let us do a quick recap of how we can package vast lengths of DNA into each nucleus. Firstly, we've learned that DNA strand is coiled around proteins. And these proteins we refer to as histones. For every amount of DNA that is wrapped around histones, we refer to this structure, this bead-like structure, as nucleosomes. Many nucleosomes can then coil around each other to form this thread-like structure, and refer to this as chromatin fiber. DNA exists as chromatin fiber for most of its cell cycle. But just before it goes into nuclear division, for example mitosis, the chromatin fiber will then coil further and condense further to form what we call chromosomes. Now, chromosomes are a much condensed form of DNA, and this much more condensed form of DNA is much easier to organize and segregate within the cell during mitosis or meiosis. Humans have, to be specific, actually we have 23 different chromosomes. Here is a snapshot of the 23 different chromosomes. Each chromosome contains certain genes. That means to say, while we have 20,000 different genes, these 20,000 different genes are all spread across the different chromosomes. For example, chromosome 9 over here contains the gene that controls our blood type. Chromosome 16 over here contains a gene that controls hair color, and so on and so forth. But guess what? Humans have two sets of the chromosomes, these 23 chromosomes. That means we have 46 chromosomes in total. We say that this is the diploid number of chromosomes, while half that number, we refer to it as haploid. So now, most of the cells in our body have a pair of each chromosomes, if you notice over here. We refer to each pair of chromosomes as homologous chromosomes. Now, homologous chromosomes, as the name suggests, homo means same. They are largely the same. They have largely the same length. They also have largely the same genes. Okay, if you can see here, the different stainings shows you where all the genes are. The location of the genes are also the same, and the centromere location is also the same. Here we don't see the centromere region, but let's say if we can, the centromere regions are also the same. But I say largely the same because they are not quite identical. If we were to compare the same gene on both homologous chromosomes, you will find that they may not necessarily be exactly the same in DNA sequence. So what do I mean by that? Let's look at chromosome 9, the chromosome that contains the gene for blood type. We unwind the DNA so that we can expose the gene. Here is a sequence of DNA again, as we have encountered last time, and refer to this sequence of DNA as a gene because it codes for a protein. We find the same gene on both the DNA strands. However, if you notice, there is a slight difference in the base pairs. On one form of the gene, we have C. The other form of the gene here, we have a G as a substitute. Now, notice that a change from just one base pair, from C to G, can actually result in a change in the amino acid coded for. If we look at this, the genetic code, CCC codes for proline, whereas CGC codes for arginine. So a change of a base pair can result in a change of amino acid coded for. And the implications are as such. If we change the particular codon that's present, we change the primary sequence of the protein. And if there is a change in the primary sequence of the protein, it can also result downstream a change in the tertiary structure of the protein. So while the protein may still serve the same function, it may serve the function slightly differently. Let's look at the example of blood type. This particular enzyme over here that the gene codes for is an enzyme that attaches sugar groups to the surface of red blood cells, which is what gives each red blood cell its blood type. Notice, however, because of a change in the sequence of amino acids, the protein that is synthesized is slightly different. They still serve the same function and can attach sugar groups to the surface of the red blood cells. However, now there's a slight difference. It attaches a different sugar group. However, because of this slight change in the sugar group being attached, we now result in different blood types. So, if we were to go back over here, we have the same gene, but different forms of the same gene. So, we will refer to different forms of the same gene now as an allele. This is one allele. This allele over here codes for an enzyme that results in blood type A. This allele over here is a different form of the same gene, but it codes for the blood type B. So in conclusion, we've learned a few things from this particular video. Number one, that most of our cells are diploid in nature, which means that it has two sets of each chromosome. And some of our cells are haploid in nature, which means that it has one set of chromosomes. Number two, we learned that each pair of chromosomes are referred to as homologous chromosomes. And homologous chromosomes are largely the same. They contain the same genes, have the same length, have the same centromere location. Location of the genes are the same. But number three, we learned that homologous chromosomes can contain different forms of the same gene. And we refer to the different forms of the same gene as alleles. We will see how alleles come in useful in the later part of this topic, and we hope to see you again in the next video.